Welcome. In this video, we'll walk you through NeuroAlign, a dynamic dual-stream framework that aligns perception and cognition for zero-shot brain-to-image retrieval. This work is presented at ICMR 2026 by our team from the Shenzhen Institute of Advanced Technology, Chinese Academy of Sciences. Let's start with the problem. When a person looks at an image, the brain recognizes the whole scene in about 300 milliseconds. From those brain signals, we want to perform visual decoding, and it comes in two flavors. Image retrieval, where we rank candidate images by how well they match the signal, and image reconstruction, where we generate the viewed image directly. But there's a tension here. Human perception is remarkably efficient, while machine decoding is still a bottleneck. So the question driving this work is simple. Do existing methods overlook key mechanisms of how the brain actually processes vision? We argue that they do, and the next slide shows why. The biology points us to the answer. Human vision isn't a single pathway, it's two streams working together. The bottom-up perception stream, shown in blue, carries low-level features, edges, color, and luminance, from the retina up through V1 to the cortex. It decides what to focus on. The top-down cognition stream in red runs the other way from high-level areas like the prefrontal and posterior parietal cortex, imposing semantic priors, what we expect and what an object means. It decides where the meaning lies. Recognition is the interaction of both. Yet most decoding models collapse everything into one pathway, and that single choice is the root of the problems we're about to see. Here's the core insight, made concrete with this zebra. Most existing methods are clip-based, compressing the brain signal into one feature vector that has to carry both what the image looks like and what it means. That creates a trap in two directions. Drop cognition and keep only perception on the left, and a striped zebra collapses into a prison uniform. It looks right, but it's semantically wrong. Drop perception and keep only semantics on the right, and the concept zebra stripes savannah retrieves a barcode. Semantically, it's stripes, but it looks nothing like the animal. One vector can't be both. So our guiding principle is this. Decode the brain the way the brain encodes, separate perception and cognition, then coordinate them. Concretely, forcing one unified space causes three conflicts, each a mismatch between what the brain does and what the model does. First, a perceptual conflict. Under rapid serial visual presentation, the brain responds to salient low-level cues, but the encoder extracts global semantic features, so the model ignores the saliency the brain actually processed. Second, a semantic conflict. The brain uses top-down priors to filter sensory noise, while the model projects noisy EEG straight into visual space, unconstrained, retrieving things that look similar but mean something else. Third, an optimization conflict. The brain resolves perception first, then semantics, but existing methods optimize everything simultaneously and statically, forcing meaning before shape, so the gradients fight. These three conflicts map onto three modules, VSE, SGA, and DLA. Here's the full NeuroAlign pipeline. The principle in one line, decouple first, then coordinate. We align three modalities in a shared space, the EEG, a visual saliency map, and an LLM-generated caption. Three modules do the work. VSE handles perception, aligning the EEG with salient visual features. SGA handles cognition, using LLM descriptions as semantic priors. And DLA handles optimization, scheduling perception before semantics. Let's take them one at a time. First, visual saliency extraction, which makes the model look where the brain looks. It has three biologically motivated steps. Rapid saliency detection uses Sobel edges plus luminance, contrast, and color, mimicking the orientation-selective cells in V1. Center surround antagonism uses a multi-scale difference of Gaussians to suppress non-salient background. And finally, the saliency map becomes an attention mask applied to the image, yielding our perceptual target. You can see it on the right with this antelope. The original is full of distracting grass, but after VSE, the attention heat map locks onto the animal's body and contours. The key point is at the bottom. VSE is an attentional filter, not a preprocessor. It discards redundant pixels to raise the signal-to-noise ratio, exactly what the bottom-up stream does. Second, semantic guidance alignment, which uses language as a semantic anchor. EEG is too noisy to learn stable concept relations on its own, so SGA borrows the well-structured topology of a language model. 3 steps. Build the prior, where blip2 captions plus clip give a semantic similarity matrix P, read the brain, where the EEG encoder gives an EEG image distribution Q, and pull them together by minimizing the KL divergence between P and Q, so Q inherits the language structure. Look at the TS&E on top. Before alignment, the categories food, animal, clothing, and tool are completely tangled. Without SGA, they're still blurry, but with all modules, they form 4 clean, well-separated clusters. That's the structured language prior rectifying the noisy neural signal, guiding by category, not just by pixels. Third, dynamic loss adjustment. Perception first, then meaning. Static weighting treats perception and cognition as simultaneous, which violates the brain's temporal order. DLA instead reweights the losses in real time using gradient dynamics. Early in training, it puts higher weight on the EEG image loss to build a stable structural foundation. Later, the weight shifts to semantic guidance once perception is grounded. It's gradient norm balancing that produces a curriculum automatically. The curve on the right shows it. Perception, the blue W1, falls as semantics, the green W3 rises, and the bands are tight, with a standard deviation under 0.1. The model discovers this perception then semantic schedule on its own and reliably. Now the results. This is 200-way zero-shot retrieval on Things EEG2 against all major baselines. In the intrasubject setting, the top block, NeuroAlign reaches 48.1%, top 1 on average, a new state of the art. But the more telling number is below. In the intersubject setting, where we leave one subject out and test on a completely unseen person with no fine-tuning, we get 14.5% top 1, again the best across the board. That gain on unseen subjects is the strongest evidence that we're capturing a generalizable principle, not overfitting to one brain. To check that this isn't specific to EEG, we ran the same protocol on Things MEG, a different modality. Same story, 26.5%, top 1 intrasubject and 3.9% intersubject, outperforming the baselines in both settings. The dual stream design transfers across recording modalities and across people. Qualitatively, here's what the retrievals look like. Notice the top 5 results don't just get the category right. They preserve color, contour, and layout. The cheese, the popcorn, the antelope, the retrieved images are visually faithful, not merely semantically correct. That's the VSE module doing its job, grounding the decoding in what the brain actually saw. The ablation tells us which part matters most. Removing VSE costs about 6 points, removing SGA about 9, but removing DLA collapses top 1 by over 23 points, by far the largest drop. The headline, "The real bottleneck isn't adding modules, it's coordinating them in time." On the right is the prompt study. Richer isn't better. A concise prompt, this is a photo of beats complex descriptions, beats labels, and beats no text at all. Under RSVP, the brain sees only the most salient content, so the prior has to mirror exactly that. And the gains hold across BLIP2, LLAVA, and Quen, so the method is model agnostic, driven by the guidance strategy itself rather than any specific LLM's knowledge. Finally, 2 checks that we're decoding the right thing. On where, the learned saliency concentrates on the occipital and parietal regions, the anatomy of the bottom-up perception stream consistently across all 10 subjects. On when, VSE drives a rapid early surge between 100 and 500 milliseconds, while SGA sustains accuracy past 500 milliseconds, anchoring against concept drift. The model's spatial and temporal behavior both line up with known neuroscience. To conclude, NeuroAlign offers a decoupled paradigm, framing brain decoding as dual-stream retrieval rather than a single projection. 3 coordinated modules, where VSE grounds perception, SGA guides cognition, and DLA schedules them in time, and state-of-the-art results with strong generalization, all in a lightweight real-time model with just 1.17 million parameters. We'll be honest about the limits. We lean on pre-trained clip and LLM priors, which may reduce decoding to matching within a fixed space, and static prompt templates constrain conceptual diversity. The natural next step is dynamic semantic guidance that pushes BCIs beyond static feature spaces. Thanks for watching. If you have questions about NeuroAlign, the contact details and full paper are on screen.